A little bit of the outline today. I'm going to start giving a quick presentation of the problem in Powell, scale insect taxonomics with the Florida Department of Ag is going to talk about the biology and ecology of these insects and what it is. Dr. Isaac Esquivel, our agronomic and forage crop extension specialist at the North Florida Research and Education Center, is going to talk about the management in pastures. And Dr. Difen Mu is our assistant professor in entomology at the Everglades Research and Education Center, and it's going to give you an update about sugarcane. Okay, let me talk a little bit about Pasture Mealybug and why it has been a major topic of conversation among different commodity groups in Florida, especially cattlemen in the sugarcane industry. So pasture mealybug is a small insect. It's different from other insects that affect citrus and ornamentals, for example, because it specializes in grasses. It can feed in any type of grass, including forage, agronomic crops, turf, and weeds. The origin, we know a lot about it from Australia, but bringing here to the United States was first detected in Texas in 2025. Was probably there a little earlier. Our colleagues at Texas A&M had mentioned that there was some suspicion of the mealybug before that, but was first detected in Texas last year. And since then, it has really spread. We're over 70 counties in Texas. In May 2026 was first detected in Florida. It was also likely to be here before. Some of our ranchers had mentioned pastures looking off as early as fall last year. And we officially first detected the last week of May in Florida when FDACS was able to confirm the presence of the pasture mealybug. First week of June, we jumped right into this. We put together a task force between University of Florida and Florida Department of Ag to understand more, to investigate more, and to also help our producers. Now we know that it's spread to over 15 counties in the southern part of Florida. We have not detected as of recently, have not detected in north part of the state. Why it has become a major issue, one of the problems is that is quickly spreading. We're finding more and more, and it's very quick to make damage in some of the crops. We have a lot of unknowns. We have a lot of unknowns about the biology and about the ecology and control here in the state of Florida. We don't know much about insecticide efficiencies, so we're putting together a lot of trials, putting in the field so we can inform you better about insecticides to be used. We're going to talk about those today. There is also label restrictions. You know, the label is the law. We need to follow the guidance of the label. There is a lot of limitations because we didn't have until recently anything labeled, but there is a lot of progress in that end. Now we have the label, we need to understand the efficiency. It also a problem with habitat, and the habits, the bottom part of the canopy, the thatch, and the soil. So it's also very hard to reach with insecticides. So we're investigating different managements that we can apply. And as I mentioned before, it goes through different grasses. We have in sugarcane, as the pictures in the left here, we have limper grass as an example of forage grasses that it affects in the bottom right, and it can also affect turf as an example here in the top right. We have not had a lot of problems in turf, so turf is not one of the major topics of discussion today, but it has the potential to affect turf grasses also, so we need to keep an eye. A lot of people have been asking what we've been doing so far. As I mentioned before, we jumped right into this problem, but things in science move slow because we need to make sure we have good results before we can recommend things. So people get a little bit frustrated. What can I do? You're not giving us recommendations. Yes, but we're investigating a lot. Dr. Julian Beslin is working with colleagues in sugarcane, now with colleagues in Belgrade, focusing on sugarcane. Dr. Nicole Quinn and colleagues in Indian River are assessing susceptibility of different grasses and also some tolerance of effectiveness of insecticides. Dr. Brent Sellers, Isaac Esquivel, and myself, we started recent projects with insecticides, and we're thinking about some other management trials we're putting in the ground. And Dr. Aaron Paul in the group from FDACS had also worked a lot in the identification, trying to understand the spread of this bug and helping also our colleagues in extension to understand this. There is a lot of information out there. There is a lot of things coming up. We're getting many calls, many questions. We're getting requests for interviews. I know some of you are here today, so I encourage you to keep an eye on our website, Pasture Mealybugs. You can go through the short link here in the bottom of the screen, go.ufl.edu/Pasture Mealybugs, and is going to take you to this website. We're posting the most important, most recent information, and we have also blog posts in the bottom to keep you updated. We also have a new EDIS coming out. For those that are not familiar, the EDIS is our extension technical information publications. This has been recently submitted, is under review, and we hope that in the next week or so, it's going to be available in our website. So keep an eye. There's a lot of information, a lot of good information there. And for those that like podcasting, take a look at our Cow Talks podcast. We have 2 episodes about Pasture Mealybug with more details, both featuring colleagues from FDACS and also the experience from Texas. We are learning a lot with them. We're also learning a lot with our producers. So for those that are here that have been testing, have been trying different strategies to control this, we'd love to hear from you in our website or directly through our email forages@ifas.ufl.edu. You can reach out to us and tell us what you've been doing, because this is important for us to understand and narrow down some management practices that might help. And with that, I'm going to invite my colleague, Dr. Aaron Powell, to take over and present to us here about the insect biology and taxonomy.
Thank you for the introduction, Marcelo. My name is Aaron Powell. I'm the scale insect taxonomist with the Department of Agriculture and Consumer Services here in Gainesville. And today I'm going to talk a little bit about the biology of pasture mealybug. So pasture mealybug is a mealybug. We have over 2,000 species of mealybugs in the world and about 80 in Florida alone. And these are true bugs. So they're in the order Hemiptera. They're related to things like aphids, spittlebugs, chinch bugs, cicadas, all of these insects that all share the same trait that is a piercing sucking mouthpart. So mealybugs have long mouthparts that they use to tap into the phloem of the plant and eat the sugary kind of nutrient-rich juice that plants produce. So this mealybug in particular is responsible for what is known as pasture dieback. So this term was coined back in the 1920s in Australia when they first had their outbreak of pasture mealybug. And this mealybug is a grass specialist. So it only attacks grasses in the family Poaceae. And the damage usually starts as a yellowing or a chlorosis. And then it will turn to red or purple in a lot of the grasses that it attacks. And then eventually those leaves senesce or die. So this is an example here of thin paspalum grass that has turned red, and you can see that one of the mealybugs is being pointed out there, where some of them are turning red and purple. Other grasses we're finding don't really go red. They just turn yellow before the leaves die, and this is the case in sugarcane and things like guinea grass, which also goes by like mombaso grass in the kind of pasture hay world. So just some examples of this reddening that we see, and the left is some Haemothria lymphograss that's gone red, and you can see kind of towards the bottom of that picture, this field was very obviously red just from driving by quickly on the highway. And then on the right-hand side, we have that kind of ditch grass, that thin paspalum, which was an early indicator that we had pasture mealybug around, because to the top of the photo, you have Bermuda grass and Bahia mixed, and it was actually a pretty heavy infestation there, but it was that really bright red stuff in the ditch that like kind of led me to find that infestation. So just another example of that same site where you have that really red in the ditch, and then your actual forage that's being grown for hay is also heavily infested, but not quite as obvious until you get up close, and then you start to see those white fuzzy insects. So a little bit of history of pasture mealybug. Like I said, it first had a big outbreak in Australia in the 1920s, and then it was kind of quiet for a while. And long story short, the species wasn't even described back then, but then it got described from specimens from Pakistan and then from India. So we think that it probably is native to South Asia originally, but it really is known best about its biology and damage from Australia, because it's been such a big problem there. And it really acts like it's invasive in Australia because it's so pestiferous there. So we first started seeing pasture mealybug pop up in the New World around like 2019, 2020, where there was a couple of reports from Puerto Rico and Barbados. And then in 2025, Mexico found it for the first time in sugarcane in Cancun. And then in 2025, Texas first reported it in the United States. And then we have had subsequent reports in Louisiana, where it's a big problem for their sugarcane. And we had our first report in Florida. The first specimens that I received were May 27th, 2026. And there's a little bit of kind of scientific background here with some confusion with the different variants that might have been spreading around the world. But in general, we're calling this, pasture mealybug, and this is kind of the history of it. So as I said, first report, May 27th, 2026. But as Marcelo mentioned, we've heard that there probably has been, some damage being noticed for at least, the beginning of this year and maybe into the fall of last year. So FDACS quickly put together a survey, and we found it in 9 counties within the first 4 days of surveying. So it was clearly here for a little while longer than anyone had realized, and had already been spreading for a bit. And our current distribution as of today, the last county that came in as far as a new record was DeSoto, which was Friday. So we are at 16 counties now in Florida, and that's generally kind of our central south region there. Highlighted in gray are our positive counties. So our known host plants are pasture grasses, sugarcane, those kind of weedy roadside grasses. We have not found it on turf in Florida yet, nor have we found it in sod. There are reports from Texas that it has attacked St. Augustine grass, so that is something that we're keeping an eye out for, but right now we have not found it in kind of purpose-grown turf grass. So some examples of some damage to limpo grass you see the deep red color, which I know that limpo grass turns red naturally as it ages, but this goes extra red, and you can see all of the biomass that's dead here from the leaves senescing, dying. And again, pasture mealybug and sugarcane has a little bit of a different look to the damage. It doesn't really turn red, it just stays yellow, and then the leaves die. So here's our current Florida host list as of July 2026. So we've found it on about 18 hosts in Florida now. These include your purpose-grown forage grasses such as stargrass, bahia. We do have it on Bermuda. That hasn't made it into this list yet, but limpo grass, and then sugarcane, and then some of the other grasses here, the ones that have asterisks. Asterisks are native to Florida, and then a lot of these, are kind of like your weedy things that we don't really want. So like Kogon grass, it's welcome to eat Kogon grass, but unfortunately that's not something that it prefers. It's just kind of spilling out onto some of these other different grasses that are on the ditch sides. So detecting and looking for pasture mealybug, females and the older nymphs have 2 different types of wax. And to kind of give you a little bit of idea of the biology of mealybugs, the mealybugs don't look like typical insects in a lot of ways. Your adult females, which are shown here on the right as an adult female, and then central, the big one on the left, is an adult female. They never get wings. They kind of just stay like this soft body. They almost look like the nymphs or the immatures. They just kind of get bigger over time, but they never have like this big transition. Like you think of a lot of insects where their larval stage or their nymphal stage looks completely different than their adult. But in mealybugs, that's not really the case. So we call them neotenic, which means that they kind of just like look like babies throughout their entire life. But these are our reproductive females. And the identification of mealybugs is complex, but basically they have to be slide mounted, and you have to look at all of these crazy ducts and pores that they use to make their wax. And that is kind of why you see these different variations in the wax types, is because they have different little structures on their skin to be able to make different types of wax. So the adult males are rarely seen. They're very short-lived. They don't feed. They only have a single pair of wings, which is a lot like the fly order. And they have these long wax tails, but they're kind of rare to see. And again, don't feed. So they're not really like a big focus for us as far as control or detection as much, just because they're uncommon. So the reports from the Australian literature say that the adult females don't feed at all. They just kind of sit on the undersides of the leaves waiting to mate, and then they go down to reproduce in the thatch or the soil. But it's actually the nymphs that are causing the most feeding damage. So you have a first instar nymph, a second instar nymph, and a third instar nymph. And then your fourth instar is your adult female. And the males have a more complicated life cycle that I won't go through. But just to give an idea of size, this is my index finger is in the center there. That's an adult female. So we're talking about very, very tiny bugs. And you can have anything from this really heavy infestation here on the left to a very light infestation on the right, where you just have one nymph present. But unfortunately, some of the data says that it doesn't necessarily matter how dense the infestation is. It's almost like a presence absence as far as the damage goes, because it seems like their saliva impacts the ability for plants to defend themselves. And then the plants become susceptible to secondary pathogens or like fungus. So the mealybugs themselves are not known to transmit any viruses or bacteria or pathogens of their own. Like a lot of the true bugs that we, know of, that they actually transmit something, but these just seem to kind of cause the plants to lose their defense mechanisms and really affect their immune system. So again, an example of a tiny nymph relative to my fingers. So just being very careful with scouting and looking for these tiny little white things in comparison to some heavier infestations and some really obvious heavy damage on a couple different species being grown for hay there. So females turn pink after mating. So they are that kind of creamy white color when they're on the underside of the leaves. And then once they mate, they turn pink and they drop down into the thatch. They are very, very mobile for a mealybug. A lot of times mealybugs kind of just sit and feed, but we find that these are able to move around quite a bit. Unfortunately, they're very leggy and kind of can get themselves around quite well. So, and because of this, we're also seeing them kind of pop up on non-grass hosts. So if there's, some other kind of weeds around pastures, they might, walk over there to deposit their nymphs, but that they're not necessarily feeding on that host. It's just because they're dispersing far and wide. So the mated females, again, increase their dispersal behavior. So you're going to want to look down in the thatch or in the soil for kind of white, waxy material, or for the adult females themselves walking around. They may also be on the underside of cow patties. These are photos from Florida, but the Australian literature had mentioned that they were under cow patties in the cool, dry conditions in the winter, but it seems like they'll go down on the underside of them regardless of the seasonality. So in Australia, they found them very deep in the soil, and we have also found them in the soil here in Florida. So the picture on the left is sugarcane thatch, and I've found females, reproductive females, like at least 3 inches deep in sugarcane thatch. So we're not exactly sure how deep they're going in Florida, but we're going to put some subterranean traps out to figure out how deep exactly they're going, so that we have an idea of that for the management side of things. So how have they moved around so quickly? Unfortunately, they move on shared equipment. They move via sprigging of infested material. And they're also moving around on, people, clothing, equipment, via the winds. As you can see, they're extremely tiny. So this is a picture of a first instar nymph on the face of a crab spider. So the mealybugs, that their main dispersal stage is that first instar. So we call them crawlers, and that's when they're kind of moving away from their mom and going and finding a new feeding site. So this is the stage that is really doing most of the movement and the spread. And some suggestions for sort of sanitation to kind of reduce the spread would be using spray bottles of isopropyl to disinfect your shoes and pant legs if you're walking through a known infested area. Avoid driving vehicles into infested grass entirely, and then sanitizing those vehicles when you're leaving infested areas. And then similar with equipment, so roadside mowing, cutting for hay, we need to be really careful with that. And we're looking into some kind of clean sprigging protocols, because we realize that having clean planting material is going to be really important moving forward, so that we're not spreading this between fields and kind of having a problem before it even begins with having to combat a whole new infested field. So there are a lot of mealybugs on grasses. As I said at the beginning, we have over 80 species of mealybugs in Florida alone, 12 genera, so the genus, the species, the 12 kind of groups of mealybugs specialize exclusively on grass in Florida. And then we have other mealybugs that are generalists, so they feed on all sorts of things. So just kind of keeping in mind that just because you see a mealybug on grass in Florida doesn't mean that it is pasture mealybug, but we do really want to be careful with being, conservative with detection, and definitely reaching out to our extension agents and to FDACS when you have a sample to submit. We would definitely want to be able to see it so that we can confirm and help you out with the identification. So on your far left there is sugarcane mealybug, which is common on sugarcane, but it's usually on the in the leaf sheath itself, so you'd have to pull it back to actually see them, whereas your pasture mealybugs are going to be really exposed out on the underside of your leaves. And similarly, the top picture and the far right, these are grass specialist mealybugs that you kind of will have to pull the plant up and actually see the crown or the roots to be able to see these. And then one kind of lookalike that we've had some comments on already is Stomatomaranx, which is a muhlygrass mealybug, and that's the one in the center bottom here. And you can see that they kind of have those crazy wax rods similar to pasture mealybug, but this is a muhlygrass specialist. It also has a really dark body. whereas our pasture mealybugs are always going to be cream or pink, depending on where they are in their life. But this is kind of a gray-bodied mealybug that's only on muhly grass, and we don't have any records of pasture mealybug on muhly grass yet. And I don't necessarily think it's going to be a great host for them, but time will tell. Another lookalike is Forages. So again, pasture mealybugs specialize on grasses. So if we are seeing things on ornamentals, so, this mealybug is really common on hibiscus, for instance, and really common on croton and kind of your various ornamentals. And it has these kind of iridescent tubes of wax that they make as well, which is really similar to pasture mealybug, but you can see that they have the dark stripes down their back. And they could be on grasses, but are very unlikely to be. So this is just one to, if you have something on an ornamental, it's very unlikely to be pasture mealybug. But we are definitely happy to take a look because we do want to be really careful. And, we know that there's a lot of mealybug diversity. So definitely want to support helping with identification and detection. So right now, we're trying to delimit the pest still. We're trying to figure out which counties have it, so that can inform management and kind of know how much acreage is affected, what we need to be doing as far as looking at management in different regions on different hosts. So figuring out just like where it is a really important first step, and it will guide research moving forward. Currently, there is no regulation of this mealybug at the federal or the state level. We need to realize that we're really out to help the stakeholders, and knowing more about this bug is critical to do so. So for reporting, we suggest mailing specimens with data. So we need to know where they're from, where they were collected, what the host plant was, because these specimens actually go into a collection. So we have the Florida State Collection of Arthropods here in Gainesville. And once they're slide mounted and put on those glass microscope slides, they get put in the collection. So they are ultimately museum specimens as well. So it's really important for us to have their data to kind of progress science. So if you think that you have a pasture mealybug problem, you can submit samples directly or also contact our helpline, which you can find on the FDACS website. If you don't have the time or the vials, we can always send out a local inspector or an extension agent from IFAS to collect samples if you send us the coordinates and give us permission to come collect. Again, we have more resources on our website there, and so you're welcome to contact me. My email is listed below.
All right, good morning, everyone. My name is Isaac Escobar. I'm an agronomic and forage specialist up here at the North Florida Research and Education Center in Quincy. And I'm going to talk to you about what we know so far on how do we go about controlling a pasture mealybug. Here's my computer's a little slow, so bear with me. Right. So how do we manage a new invasive pest? And generally, when we're dealing with new invasives, we got to ask ourselves a few questions. So one, how do you identify it, which Aaron covered pretty well with those really nice photos in the previous talk. But then how do we look for it? Then how do you kill it? Does it even matter? I think that question sort of answers itself. And then these later questions, when does it matter? When do you kill it? I think thresholds will come later down the line. Now, how do we go about looking for it in a pasture? As we saw, they are very tiny. And we have mentioned, there's a lot of what we call pasture decline. If you just look for something or see something that doesn't look right, ain't doing right, you might want to go check it out a little further, especially now that we know pasture mealybug exists in South Florida. So if your pastures appear in stress when they shouldn't be, if you see some yellowing, reddening, or purpling, Erin had some really good images of some of the injury differences between the injury in the grasses in her talk. And it's seeming like we're definitely going to want to detect it before you can see extensive reddening, because it appears like it will be a little harder to manage when we do start seeing that reddening or dying patches. You want to get on the ground, look at the leaves for adults and nymphs, and definitely inspect the thatch and around the soil surface, because the crawlers are very tiny. So if you're not seeing adults, for example, like at this middle picture here, if you're not seeing adults, or if they're at this level, they're likely going to be probably at an economic level. But if you're easily able to see adults, you're likely going to have crawlers in the soil as well. Again, Aaron touched on this a little bit, but not all mealybugs are the pasture mealybugs. There are a lot of lookalikes, and these are some images that either from myself or Dr. Nicole Quinn took looking in the field a couple weeks ago when we were putting out insecticide trials. Now, this is not interactive, but if you were to guess, how many of you would have got that these two are both pasture mealybugs and not the rest of these here? So one, just order to identify that you do have pasture mealybug and it's not something else, it's not a disease that you're assuming is pasture mealybug and end up treating for something that may not be needed. And again, going into that, also when you're looking for injury, you also can assume that it's also mealybug injury. So this is a field that Jackson County went out to as there was a worry of pasture mealybugs, and go to the field, looks like this. For those of y'all that have had pasture mealybug or have seen injury, can't really tell the difference. You still see the same. We'll see that in the next slide. So here we have some fields in St. Lucie County here on the left-hand side, and then Polk County that have pasture mealybug and some pasture mealybug injury. And again, can't really tell the difference between these two images and the Bahia fields in the last two images. So again, it's going to be important into getting in the field and looking to see if it is pasture mealybug or not. It's definitely something you cannot ignore, and this is a good aerial image from my friends out at Texas A&M. You can see here in the middle where we have a dead pasture from pasture mealybug, and the far right-hand side, I think y'all can see the mouse. We have a pretty high infestation level here where you can see visual injury throughout the pasture. And then early infestation, it's somewhat barely noticeable. It might not look, doesn't look like a healthy pasture, but slightly something looks off. So this is what you would want to be getting into and getting in before you can see it at this level here on the right-hand side, or the critical infestation level here. Now we found pasture mealybug, so now how do you go about killing it? And unfortunately, I think many of you know that on the pasture side, we already have a limited amount of products that are available for use in pastures. And it happens that everything that was currently available for pasture use doesn't look to be quite effective against pasture mealybug. Those things are like pyrethroids, say bifenthrin, or your lambdicize, your carbaryl, lanate, and dimilin, and other growth regulators. They don't have much efficacy in the data I've seen from folks at Texas or pasture mealybug. Now we are testing, Texas testing as well, a lot of other products, things like Courier, that looks good. Movento may or may not be good at control. Imidacloprid looks excellent, and we'll talk about that here in another slide. We did test that in a couple fields in Florida. Centric also looks good. Fipronil, and Texas is testing it. Haven't seen much data on the fipronil yet. And there are many other potential options that could be tested, just getting to see if we can get a label for those products. Now, we did get some recently labeled products for Florida. The first one being Safina and Scalus, which got a 2EE because it was labeled for pasture previously, just not pasture mealybug. However, it is only labeled for suppression, meaning it's not going to control the problem. So you're going to want to potentially use this if you have very low populations of pasture mealybug and not in a high population situation. And it's labeled at a rate of 8 to 10 ounces. And the reason why I haven't recommended this myself is because their data on efficacy ranges from anywhere between 15 and 61%, depending on rate, location, timing. There's some reduction in mealybug injury from what data I've seen. It's just inconsistent across multiple trials. So it's not reliable. There's not enough data for it to be a reliable option. The next one that recently got a label for Florida is Altis, and it's 24C local needs label. It's labeled a rate of 7 to 14 ounces. And this, most of the data we've seen from it was about at 6 ounces per acre, so that it's labeled at 7 to 14 ounces per acre. It does look pretty well. It's kind of just summarizing the Texas data here. And in Texas, we're seeing 40 to 78% control after 14 days after application. There also seems to be good canopy protection, but I will say that they tested it in different grass. And mostly what we're testing and what we're seeing it in is on that limbo grass. And it was all tested at 6 ounces per acre. Now, in Florida, we put out a couple trials with Dr. Brent Sellers and Nicole Quinn a couple weeks ago. This first one out in Polk County that was sprayed on July 1st. You can see here, it's a Bahia field. You can definitely see some injury there in the middle, right where that ATV sprayer is. And we applied Centric, Savanto, Transform, Carbine, Admar Pro, Movento, and then Venom. And recently had some, Brent went out and did some data collection. And to count bugs, to count mealybugs, it is going to take a lot of time, so we don't have the actual data because they got to sit in funnels and count over a series of days. So this is some observations after 7 days or 8 days, 7 days after planting. It seems that after application, seems that one Centric looks pretty well, kind of consistent with Texas data, and that no mealybugs were observed in sprayed plots. Admar Pro, which is imidacloprid, and Transform, also very few individuals were found in that plot. Movento and Savanto were a little variable, but definitely less so than, say, untreated controls. Venom, they were fine, didn't seem to work in this trial. Both adults and nymphs were found easily in that treatment. And then Carbine, which is interesting. No adults were found, but just tiny nymphs. So definitely need a lot more testing on that particular product. And in terms of the untreated adults and nymphs were found easily throughout. So it seems to be consistent data with that of Texas efficacy data that we've seen. Now we put out another trial In St. Lucie County, similar treatments, 2 less treatments here. That was sprayed on July 2nd. One centric again, transform carbine imidacloprid, Admire Pro, and then Venom. And we will get data in our next update for that particular trial. So now how do we go about management for Pasture Mealybug? Well, it seems like it's going to start with early detection. It's going to be the number one thing, or best way to get control of this before we see extensive injury in a pasture. So you're going to want to scout the edges of damaged areas. So if you already see some dead spots, don't look in the dead spot for Pasture Mealybug. See on the edges of that dead patch to some of the green foliage next to it to see if there's mealybugs in the unsymptomatic grass there. And then again, confirm it's Pasture Mealybug, check the lower leaves, thatch, soil surface, drought, nutrient stress, disease, other insects can produce similar symptoms. So you definitely want to make sure it is Pasture Mealybug. Now, it is hard. It seems to be we don't have a definite way in terms of producer consultant methods for determining the size of population when in the field. But we have come to the conclusion that if you're able to see adult mealybugs, you're likely going to be at a economic level of infestation. Okay, now, so can cutting or grazing suppress Pasture Mealybug? We believe it is going to be important, probably the most important thing, just depending on where you're at in the season. So cutting, shredding, grazing, even removing that infested foliage can reduce some of that dense canopy structure, especially in the nymphograss, where it makes insecticide applications hard to get in contact at the bottom of that canopy there. Now, with that in mind, how do we go about potentially optimizing management, given if cutting is going to be important? We do plan on testing it. Speaking with folks in Texas A&M, they have also tested it, and it does seem that cutting does reduce numbers quite well. So again, it's reducing the biomass. So if you detect mealybugs and you're ready to cut, would say probably go ahead and cut. You can remove that grass layer and then allow a little regrowth to about 6 to 8 inches. And then we can think about applying insecticide just to get that product where mealybugs would be. And that's going to offer a better opportunity in terms of managing this long term, opposed to trying to take care of it when the grass is pretty dense. Now, when you do cut, what about the hay? Can Pasture Mealybug be moved on the hay? We saw our concerns on movement. We do have a slide on that as well. But what about in hay bales? So just thinking about it, when you cut the grass, Pasture Mealybugs, they're piercing-sucking insects. They feed on plant sap. So when you do cut that grass is going to start drying. So you would assume they're going to move away, right? And Texas actually has some data that this came out a couple days ago that they looked at bales, counted Pasture Mealybugs, and looked a couple days after, this 48 hours after, and they found only about 5 to 8% remained in the cut grass after 48 hours. So it does appear like they are moving away from baled hay. So just something to think about and have in mind. Now, again, just to stress, don't help move it. So again, equipment and field-to-field spread. So say you have to cover 1,000 acres of pasture, maybe one of those pastures is infested. You're probably going to want to work in your clean fields beforehand before you go into that infested pasture, or if you do need to go in there to make an application, do a cutting, definitely want to clean your equipment before moving into a clean pasture, mealybug-free pasture. And again, here's an image just on my pant legs from just within a couple minutes of stepping out of the truck to get our trial set up down in Polk County a couple weeks ago. And so again, what we kind of know, cutting, grazing, suppresses populations. It's going to help in the long run, I think, in terms of managing this pest. It seems to like dense forage over, say, thinner forage. And we have, with it being able to be in the ground, populations are going to be likely to return once grass starts regrowing as well. And then again, stressed pastures decline more rapidly than, say, healthy pastures. But there's a lot of questions we still need to ask and learn, things like economic thresholds, best cutting grazing time. Again, looking more into the hay-associated spread or plant material spread, grass and cultivar susceptibility, because there appear to be differences just from general conversation between different grass types. And then again, how does the pasture recover? It's a lot of things we do not know yet, hoping to answer as the season progresses. So with that, this is a evolving situation. So if you have questions, feel free to reach out here. Contact info is in the top left. I want to thank Dr. Brent Sellers and Nicole Quinn for assisting in getting some of these insecticide trials out and collecting data over the next couple of weeks. And if you need more information, scan this. As Marcelo mentioned, takes us to our Pasture Mealybug website for real-time updates on any control options or insecticide products becoming available. All right, thank you.
Thank you, Isaac.
Okay, good morning, everyone. Before I start, I want to acknowledge Dr. Julian Buslin. He's the leading the efforts in sugarcane here, and this is a collaborative effort between Dr. Julian Buslin, me, and two of our extension agents, Dr. Matt Van Wilden and Christian Kramer. So pasture mealybug has been a huge problem down in here, sugarcane producing area. Sugarcane in Florida, we have around 400,000 acres in the area, and they are mainly produced in these 4 counties you can see from the maps, including Martin County, Glades, Henry, and Palm Beach counties. So this is the biggest row crop in Florida, and it's planted between September and January. And once it's planted, it can harvest every year and up to 3 to 5 years. So this is multiple year crops. So it kind of make the issue more complicated. And you can see the Pasture mealybug. The purple area is the sugarcane growing area. You can see from the map, and this starts indicating where the pasture mealybug was detected from the beginning and what we are hearing from so far. But we are hearing more detection from the muck soils. So mainly it was detected in sand soil. So it's not the main area. You can see the right hand side, but now we are seeing more reports from the muck soil. So we have to understand more how the infestation is going in the muck. So this can be a little outdated, but the areas we have here is so far it's highly infested, and we are seeing a lot of economic damages here. So sugarcane infested with pasture mealybug, you can notice the yellowing leaf. That's very similar with what's reported from Australia. It's called yellow canopy syndrome. So we can see the yellow leaf of the canopy, and it starts from the bottom part, but it progresses to the top part. And you can see this is me. I'm 5'3". So you can see pretty high up that you can still see the yellowing. And from the field, when you drive past by the field, you can already notice the severe yellowing. And some field we are seeing here from the photo, it's been about a month since the detection. Some field, it's even worse than these pictures. So it's pretty bad how it's affecting sugarcane industry. And this is what I mentioned from the yellow canopy syndrome from Australia. So they have this symptom progression photos from the paper, and this is highly resemble with what we are seeing from the field. So the midrib stays white, and sometimes you see half the leaf is yellowing, and as symptom progress, you will see the entire leaf turns yellow, and it has this gold yellowish hue. So that's something different than what we see from aphids infestation or something else. And this is the field later in the disease, the symptom progression. You can see the leaves start drying out, and the topper, like canopy part, you see more yellowing. And at the end, the right-hand side, you'll see this stage. It's unlikely the plants will recover. So it's causing huge damage, and this is it's directly connected to the yield loss. So mealybug, this is how the mealybug is observed from the sugarcane leaves. Sometimes you see sporadic infestation, but the number can be very high. You see, we observed up to 400 single leaf of the sugarcane. So the infestation can start from the bottom, but it can also move to the top part. And when the infestation level is high, we see a lot of pasture mealybug plants. And I think Aaron and Isaac, they also mentioned other lookalikes. So this is what we observed from the field. Left-hand side, you can see this is a pasture mealybug, but the right-hand side, this is some other scale insects. So I think that's also important to really know what you're looking at, because this scale insect is not really causing the same damage as the pasture mealybug. Okay, so pasture mealybug is generally associated with yellow canopy symptoms, but we yet to really do some experiment to identify what's really causing and how many insects can cause this yellowing. Because from Australia, at the beginning, the yellow canopy syndrome, they were thinking more about the nutrition deficiency or something else. So in later period, they associate this with pasture mealybug. So it's still not 100% certain it's caused by the mealybug, but what we are seeing is highly associated with pasture mealybug. So mostly what we are seeing is the lowest leaves that's alive, that's more infested, but when the infestation level is high, the upper part is also infested. Okay, and As I mentioned, what we are seeing is mainly in the sand soil. That's the mineral soil. So it's around 115,000 acres are high risk. But as we hear more detection from the muck soil, so we had to see how much infestation is in muck soil and how bad it is infesting the sugar canes in muck soils. But with the infestation, the up to 50% loss is expected from what we are seeing from Australia or Louisiana. And we don't know any varietal effect yet. So what we are doing in the field, for the scouting, we usually do the direct count from the leaf. So you can see, so usually we have a counter, so we can count how many mealybugs are on the leaves. But so that's more the direct count. So the number can be not 100% accurate. And another method can be used is taking the leaf samples, put them in a bag with alcohol and bring that, and we wash with alcohol and filter them, and we do observations under microscope. So this can give us better idea in terms of the actual counting, but this is more time consuming. So now we are doing more field observation, direct count. And the roots can be also infested, and post-harvest residue can be also a risk. So this is what we developed for the scouting in the sugarcane field. So using the direct leaf observation, so count directly from the leaf. And what we're seeing now is pasture mealybug infestation tend to be more on the edge, but actually what's happening in the entire field, it's not necessarily the same as the edge. So we suggest do a zigzag or W pattern across the field and do 5 to 10 stops if possible, at least 100 feet apart. So then you can have a better idea the overall infestation in the field. And each stop inspect at least 3 plants, so 3 stalks, that's 5 to 6 feet apart. And so this is what we, based on our observations so far, this is what we think the scale level of the pasture mealybug infestations. So 0 meaning there's no mealybug observed, so there's no infestations. If you see 1 to 5 per plant, at this point with the low infestation, they tend to be more on the lower part, but lower it's part, but not the dead leaves. So you still have to check the yellow or half yellow, half green leaves, but not the leaves that it's dried out. Yeah, so here if you see 1 to 5, then it's low infestation, and later 6 to 25 insects per plant. At this point, not only the lower part, but the infestation can also show on the middle or upper part of the plant. In high, 26 to 100 mealybugs per plant. And at this point, you will see mealybugs on both lower and upper leaves. And as I mentioned, we see more than 100 and up to more than 400 mealybugs on plants. So the infestation level can be very, very high. And usually you can observe them with naked eye, but sometimes hand lens can be very useful. If you need to confirm, because the nymphs can be very small and it's easily missed if you don't really know what you're looking. And the yellowing symptoms, similarly, they start from the lower canopy of the plant and later it progresses up to upper parts. So if there's no yellow leaves, then meaning there's no symptoms, and low, very low, up to 25% of the plants, very low to low, 25%-ish, and moderate 50, and then high 75 to 100. So as now we see a very high infestation level, and sometimes they don't really have a leaf that's alive. Yeah, so in this point, that plant is dead, then so that's the plant is not recovering. So for the management perspective, now we have the excuse me. Can you still see my PowerPoint?
We're seeing your regular screen with the presentation.
Okay, now it's good.
Cool. Now it's good.
Okay. Yes, sorry about that. So now Sivanto is approved under Section 18 crisis exemption. So it's ongoing, 7 to 14 fluid ounce per acre and maximum 2 application per year. So this is already approved, and I believe they are Many acres are already sprayed with Sivanto. And we have another Admire Pro is just approved on July 14th. So that's effective for 2 weeks from July 14th. So this is also available for spray. And because the Admire Pro, the limitation of the insecticide availability, there are 11 more additional formulations of imidacloprid is also approved. So these two, Sivanto and Admire Pro, they are approved as of now under Section 18 crisis exemption. And for the quarantine exemption is still ongoing. But hopefully it will approved soon. And this is one of the insecticide efficacy result we put out. So far we have 2 insecticide trials. So this is the first one. The location is Port Mayaca. So we have 8 treatment, including untreated, for the Pasture Mealybug control. So 7 days after treatment, we don't really see significant difference among the treatment. But 14 days, we see some of the treatment, especially the Movento, showed low numbers of Pasture Mealybug. And 21 days after treatment, we see the Sivanto and Movento, the combination showed the lowest number of mealybugs. But still, they are not killing all the insects. So it's hard to say like later, like how they might like grow as a population. And we also start counting the yellow leaves. So how we do it is we count the yellow leaves and the number of green leaves. So we ignore the dead leaves, dried leaves, and we count the yellow leaves and the green leaves. So far, there's no really significant difference among the treatment. And we also don't know if the yellowing is recovering, even if we kill the insects. So I think that's something we also have to figure out. But so far, the Sivanto and Movento combination showed the best effect. And Warrior, they have the highest number within the treatment. It's almost similar with untreated. But with the Movento treatment, we observed some phytotoxicity to the plants. So it's called dead heart. So you can see the left-hand side, the photo is showing at 7 days after the treatment. So the growing point, the middle, the newest leaf of the sugarcane is dead, so it's easily pulled out. That's dead. But we observed some recovery on the 4th week day, so it might recover, but you can still see the leaf, like the edge is cut. So we don't know how this can, so this plant, how much yield they can produce. So we had to figure it out. But we see some phytotoxicity from the Movento and somewhat recovered. And this is another insecticide trial in Indiantown. So this one, we have 10 treatments, including the untreated. But this one, so this site, we had higher infestation overall than the first trial. But we don't really see the difference between the treatment and these plants. I don't have the data now, but after 3 weeks, it's still similar. So, and the plants are in really bad shape. So what we are thinking is maybe at the beginning, the infestation level was too high to get any control of the mealybug. And the plants at that point, it's not really recovering. Yeah, even if we spray. Yeah, so that's something we have to consider. So maybe at the early point, so at the low infestation level, if we can spray, then that will give better efficacy. So from here, what do we go from here? So we are still working on to identifying effective conventional and biological insecticide for pasture mealybug management in sugarcane. So far, it's been challenging, but we are working on it. And we need to see if there is a varietal difference. So we're gonna work with the growers and see if there's any difference in their susceptibility among the commercially grown varieties. And we need to know how much sugarcane yield is associated with this pasture mealybug infestation and the yellowing, especially what we are seeing, the yellowing is not really recovering after the infestation, even if the number is lower. And we want to also quantify the symptoms and to determine the spatial and temporal variation. So we are planning to do some drone testing. So we are working with the other faculty in the station to see if we can do something so we can scout from the drone and then correlate this to the real number of infestation and the yellowing level, and also test some plants material. And to evaluate or identify the natural enemies that's effective for mealybugs control, and understanding the physiology basis. So what's actually causing the sugarcane yellowing, and like if there's any nutritional or biostimulant or anything that can help with the yellowing. So like to recover or reverse the symptoms. Yeah, so that would be our priority. Okay, so with that, I would like to thank everybody involved. Also, the Aaron helped us to identify the mealybug, and Mike Alert, so he's working on the Section 18 approval, and we are in close contact with researchers from Louisiana. So we meet every week to discuss about their research and what we can learn from their trials. And sugarcane growers, they've been very helpful to let us do the trials, and we're all on this together. Yeah, so with that, please feel free to reach out to me or Dr. Julian Buslin if you have any questions. Thank you.
Thank you so much for all the presenters. Let me start with the Q&A, and I'm going to call on Isaac, Difen, and Aaron to respond. I'm going to start with one that was there are multiple questions about the same topic here, which is what have we seen it in other things? We have questions about Congo grass. I mentioned Johnson grass. Aaron mentions mud grass, also sorghum Sudan and other pastures and weed grasses. Aaron, can you elucidate a little bit for us? Where have you seen the mealybug? And what is attacking so far?
Yeah, so there's no reports on like purpose-grown sorghum at this point from anywhere in the world. There is a report from Johnson grass in Mexico, which is the same genus. So I guess there's potential. There are some like other reports from other places historically that it could potentially go on rice, and there are some like unsubstantiated ideas that it could go onto corn if it's near a close to an infested pasture, but it's probably not a preferred host. So what we're seeing is that, if you have heavy infestations, there's a lot of like spillover into like the ditch areas of, random like weedy grasses, and you getting onto these like invasive things like Kogon or smut grass. But first of all, it's not a good biological control option for invasive grasses because it's such a problem in everything that is purpose grown forage. But it does seem to be, more of a problem in these dense, like, monocultured, forage crops.
Yeah, I think there was one report in sorghum Sudan from Texas. So that's a sorghum hybrid with Sudan grass.
Okay.
Now, also on the sorghum, remember that sorghum aphids are a problem. So if you see bugs on sorghum, it's not necessarily pasture mealybug. That's why the identification is very, very important. There was a lot of questions about burning here. Isaac, either of you would like to mention anything about the burning as a strategy to reduce population?
Yeah, I'm just going to comment too. Stephen just posted that they did have an infestation in a grain sorghum field where there was worry about it getting up to the heads. So sorghum definitely can potentially be a host from Texas. And then in terms of the burning, we've all thought about that, but I think the issue is going to be that they do go in the soil, right? And it seems to be pretty deep. So even if we were to burn off some of that thatch, there still potentially can be survivors to reinfest. But it's definitely something to look into.
Okay. On the detection side, is there any good time of the day for detection? And also, do we have a threshold? Can we say how much, if we see mealybug, how much would be a problem when it triggers management? I think Aaron had responded to that too, or Isaac?
Yeah, I agree with Aaron. It's like I haven't noticed a difference, but definitely I would think the rain is going to affect counts. So definitely don't scout after a rain, but we haven't noticed any distinct difference in time of day sampling. And in terms of threshold for pasture side, I think that's just if you easily find adults, you're probably at a threshold level or economic loss level. And I don't think we'll ever even need one. It's going to be more of a presence absence, like Aaron has suggested as well.
Perfect. Now I'm going to merge a couple of questions here on insecticides. So we have a question on pyrethroids in terms of effectiveness, specifically in cold weather or hot weather. We had dinoterifan. We have sulfoxaflor also. Any comments on either of those insecticides from Isaac or Difen?
Yeah, so the pyrethroids, at least from what I've seen, we didn't test them. Generally, pyrethroids don't work well for mealybugs, or it didn't seem to work well in the Texas data. And the other two, they're both neonics, and we are testing those, and both do look pretty good in terms of control, at least on the pasture side. They may be labeled on the sugarcane side.
Yes, so For pyrethroid, we have the Warrior, so that's the least effective one from our insecticide trial. And because they are mostly contact, I believe, but because the wax, so maybe the contact might, so it's the systemic will be better. Yeah. And then, yeah, so Warrior, it's not that effective from our trial.
Thank you. I'm going to add something about the insecticides, and many people are asking, like, why are you not recommending more? So there's two levels of that why we're not recommending. One is because we need to follow the label for the official recommendations. That's very important. But we're also trying things that are not labeled because potentially we can get those labeled. The second side of the insecticide trials is that we don't want to recommend anything that is not working well, because if it's not working well, you have two things. First of all, you are wasting money. Second, you could be damaging beneficial insects. You could be, especially with the broad spectrum contact insecticides, you're going to kill everything. And that is definitely not a good thing, because you may be killing some of the insects that can help in biological control. With that, I think there was a question there on biological control that Erin said that she would take it.
Sorry, can you repeat that, Marcelo? I'm talking to chat. I'm doing many things at once.
No problem. So biological control, is there any work being done? What do we know about biological control?
Yes, so FDACS is jumping right into testing some biological controls. We are, looking for potential agents in the field actively, and also trying to test some of the more like commercially available things. We don't have any preliminary results on that yet. And IFAS is also working on biological control with Dr. Nicole Quinn down at the IREC in Indian River. So we don't have any data on that yet, but I will say that, ultimately, oftentimes with these like early, invasive species invasions, we rely a lot on pesticides at first, but then we find that these species kind of settle in over time as the natural enemies find them, recognize them as a potential tasty food source, and that, pesticide use can be kind of tapered out as the natural enemies pick up. So ultimately, like, that is the sustainable plan moving forward. And we, the working group between FDACS and IFAS is definitely getting right onto that. So.
Another question on potential application options. Neem oil, has it been looked at?
I'll let Isaac
Yeah, so neem oil, we don't have it in our insecticide trial, but from Louisiana data, they have tested, but it shows moderate efficacy, I would say. Yeah, it's not, but they are testing, and then,
Yeah, I was saying we haven't tested it, but I think it would be worth looking at.
Okay, Defan, we have several questions here on sugarcane. And by the way, folks, I'm trying to keep up with all the questions. I'm writing them down here on a document. Things that we did not answer today. Right now here in the next few minutes, we're going to write a blog article responding to all this question, posting also the information, the website, the webinar information. Defend, there is a couple of questions about burning or detaching for sugarcane versus green chopping, and if anything have seen in reducing the population. And if you can respond that, there was another one on sugarcane. I'll find it here.
Okay. Unfortunately, I don't have the answer for that question. So I think it's this stage, it's a lot of unknown. So we have to figure that out. So now the sugarcane is more at the growing stage. So this is like summer, that's where they grow. And then, so I think the burning and chopping, it will be later in the season. We will have to do some tests to figure that out.
Okay. And is there any physiological stage of the plant, maturity stage of the plant, that would be more susceptible?
Also, we don't have the answer to that questions. Yeah, so that's really important questions we have to know. And if like the stage of the cane, or we're also thinking the more stressed cane, maybe it's more susceptible, because usually the canes in sand soil, they are more stressed than the muck. And also when they were planted, or if this is plain cane or rotting cane, I think all of this can be important in terms of how they are susceptible, or they can withstand some of the Pasture mealybug infestation. So yeah, we have to do more research to answer these questions.
Thank you. Isaac, flooding. Would flooding be an alternative to reduce the mealybug population that is in the soil?
Flooding in pasture?
Pasture flooding. For us in North Florida, sometimes it's not common, but in South Florida, we have a lot of pastures that go flooded during the summer. And also have sugarcane also could be flooded.
Yeah, that's a good question. But so in literature, it's reported in rice, right? Which is flooded. So it's kind of interesting.
Yeah, so usually rice is flooded and they can suppress the wireworm population, that's soil dwelling insects. For the next crop, let's say if it's sugarcane, then they have the decreased wireworm infestation because of flooding. But in sugarcane, usually they are not flooded, but I believe the industry, they are trying to also look into this. And I am also curious to know if they can help. But I don't know how, because they can survive in the soil. And if that's deeper, I don't know how they will react the flooding or for how long. I think that would be important because also we don't want to kill the plants. That's something to consider. Yeah.
Okay. Yes, we're also wondering if it would help, especially on the limpo grass, because limpo grass go underwater, and we had a very dry year recently, so could have that exacerbated the effect of the mealybug. We are not sure about that yet, but those are very important observations. So if y'all have any information to share with us from your observation, that's how we build more knowledge and narrow down our management strategies too. I have one on taxonomy here, Erin. Some people are asking, do we have two types, type A and B? And I think this is related to that different instars, so different phases of the insect. If you can respond that.
So we have, certainly different nymphal instars where we have a first instar crawler, a second instar, a third instar, and an adult. And then as far as the kind of types of variants, if you read my pest alert, there's some mentioning of these early observations of potentially having two variants, and the Australians have also reported similar, but research on that is ongoing. We're working on that here at FDACS with our collaborators in various parts of the world. So.
Okay, one more question here on sugarcane. Well, let me just go back before we get back to the management. Erin, anything on viruses? Do they transmit any viruses that are known of?
So Caroline Hawkswell's group in Australia, she's at the Queensland University of Technology. They've done the most work on that, and they have never found any evidence of these actually transmitting viruses or pathogens of their own. All they have found is that, their feeding disrupts the immune system of the plant. So it disrupts the salicylic acid pathway and then kind of opens up the plant to a secondary, infection of like fungus, for instance. And that's ultimately what causes the death of the plant. But we certainly should be looking at into that in the United States as well. I mean, the damage in Australia is very similar, where they have, their pastures go red and their sugarcane goes yellow. And we don't really have any reason to think that there's a virus or a disease here in the US, but we should certainly replicate those experiments here just to be sure.
Okay, there's a few questions about life cycle that I skipped here. We're going to have more information about the life cycle in our EDIS publication that is coming soon, and also on the podcast. I'm going to just skip those. And also, one of the things we don't know much about how the life cycle goes in Florida. We know the general direction of the life cycle, but that's something that we are still investigating. Now, I have a question here on sugarcane, but I think I'm going to expand it for pasture as well, so Isaac can defend. Harvesting and spraying after a harvest, can it be a strategy to reduce populations?
I guess I'll go first. Yeah, I think that's going to be an important strategy on pasture. On the pasture side is to cut, and if you're ready, whether it's for baleage, hay, whatever, to then go ahead and apply insecticide.
Also, I'm going to add to that response before the fan talks about sugarcane. We had seen, so we have not tested, so very important, we have not tested, but we have seen some producers that baled the areas that were infested, and then they have not found mealybugs in the hay. This is kind of, it makes sense because the insects are feeding on the sap of the plants. If the plants are dry, the insects are not able to survive. So this could be also a strategy, and then spraying afterwards. This is one of the things we're interested in testing. Defend, what about sugarcane harvesting and then spraying?
Yeah, I don't know. I don't have an idea because I think we have to test and see how that will work.
So we have a lot of questions here, but this is important, and remember that those questions help us evolve. Now we have a question here from our colleague Lance regarding buprofenazine. Sorry, I normally handle more herbicides, not insecticides, so some of the names are not common to me. But Isaac, this is an insecticide that is used on ornamentals. Could it be effective? And is effective on mealybugs? Could it be effective on pasture mealybugs?
Yeah, so that's a growth regulator. It hasn't been really any data tests on growth regulators. One, just because you would need them. Say it will kill them as they are going from instar to instar. So generally they don't have adult efficacy, which is kind of the reason why don't kind of test them, it's because they'll still have the adults. But that is a potential option to test.
Yeah, for us, we have included courier in our second trial, but unfortunately, the second trial, the overall efficacy is just not great due to the high infestation level. So we didn't see any result.
Fantastic. Okay, I think we answered most of the questions. If I missed anything that is important that was not answered, please type it there. I've tried to go through most of the questions that we have here. There was one more here. If mealybug will survive on sugarcane residue after harvest and then burning. So I think this is a positive, because we also see both sugarcane and pasture residue after harvest. Harvesting helps controlling it in pasture, reducing the population, but it can stay in the residue in the stubble there. Erin, any last comment that you want to share with our audience here?
I don't think so. I think that just essentially, we have a new invasive species on our hands, and we're have a good working group that is kind of coming at it from a lot of different angles, and we're hopeful that this is going to behave like a lot of other invasive species, where it is kind of explodes at first and then settles in over time. As the natural enemies find it, and as we come up with some new control and management strategies, and we will continue to communicate with the stakeholders on that, and also just kind of reiterating that we want to confirm the pasture mealybug infestations and continue to delimit that. So send specimens in for us to be able to slide mount them and identify them.
Isaac, any final comment?
No, I would just say reach out to us if you have any questions. And just say that we've been dealing with it for just about a month, and it does take time, so have patience for reliable answers to a lot of these questions, but they will come.
Difen, any last comments on the sugarcane side?
Yeah, so same here. I think it takes time, but we are on this all together, and hopefully we'll find solutions soon.
Awesome. Folks, I appreciate everyone participating today, and our speakers as well. Thank you for being here. Remember, there is some resources there for more updates. In current updates, we're trying to keep this website updated. Pasture Mealybug, so go.ufl.edu/Pasture Mealybugs. And if you have more comments, questions, ideas to share with us, reach out to us. Most of you have our direct contact, but if not, email forages@ifas.ufl.edu. That is forages.ifas.ufl.edu. And also here's a link for our podcast if you want more information. We'll probably record something soon because this is evolving. Thank you, everyone. Hope you have a great day, and keep an eye for the posting of this webinar on our social media. Thank you so much.